Assalamualaikum and a very good morning to everyone. Today I would like to present about my final year project, which is low cost ultraviolet technologies for water disinfection in-house. For getting a clean and safe water resources, microbes that contain in wastewater need to be treated. The usual pathogenic microbes that contaminated the wastewater, such as E. coli, hepatitis, polio, Giardra, and Cryptosporidium. UV radiation is the component of the EM spectrum between X-rays and visible light. UV rays have a higher frequency than visible light and a lower frequency than X-ray. Since sunlight is the major source of UV radiation that creates three main forms of UV rays, UVA, UVB, and UVC. As you can see in the figure here, UVA rays have the longest wavelengths followed by UVB and UVC rays with the shortest wavelengths. UV treatment for water is recognized as a safer and more cost-effective way to disinfect water. This makes it a practical process for treating water used for drinking and food preparation. There are no disinfection byproducts with the use of UV, which is safe and chemical-free. UV does not alter water chemistry and its constituents, such as pH, taste, odor, or color. UV has been used commercially for many years in the pharmaceutical, cosmetic, beverage, and electronics industries, especially in Europe. Ultraviolet light is increasingly being applied instead of chlorination for disinfection of both drinking water and wastewater in centralized treatment plant because it is effective at inactivating protozoans and does not produce disinfection byproduct. Also, ultraviolet disinfection systems are used in many different applications ranging from the purification of drinking water in individual homes to disinfecting water supply of entire townships to industrial wastewater treatment. Next, for the problem statement, in order to be able to supply clean water for human well-being, it must undergo a proper water treatment and disinfection process, since wastewater contains a lot of microorganisms that can cause illnesses to human beings. For commercial UV technologies in water treatment was available for every unit household, but in terms of cost is typically high and certain replacement part is quite hard to earn as it is not accessible in every part of the country. Thus, if affordable UV technologies for water disinfection is available, can we assure that the quality of the treatment water from ultraviolet light is as competent as the installment of commercial UV technologies? As the objective for this project, the first one is to study the low-cost design of ultraviolet technologies for water disinfection. Second, to investigate the effectiveness of low-intensity UV light for wastewater disinfection. And the last one, to show the sensitivity between bacterial types by establishing the required dose for a safe water disinfection that is adaptable with allowable limits. We're moving on to the literature review. The study reviewed literature from various scholars on the major variables of the study included background of UV technologies, treatment process of ultraviolet technologies, and ultraviolet technologies specialized component. By treating our water supply with the use of UV disinfection is not only more effective than traditional treatments, but it is also an environmentally responsible way of treating our water. Although it's taken a long time for the technologies to become widely adopted, ultraviolet has been around for a long time. In 1877, the germicidal properties of sunlight were discovered, and it was only a matter of time before people tried to apply this knowledge for practical use. Next, UV was a natural application because it was effective, does not require any storage or extended contact time, and does not leave any residual. For example, in North America, UV first found wide application for treatment of wastewater because chlorine residual is toxic to aquatic life. The discharge of wastewater into waterways had to cease by either dechlorinating the water or changing the disinfectant. The use of UV disinfection for wastewater treatment has increased dramatically in the last few years due to the impact of chlorinated organics from sewage effluent on receiving water. In brief about the background of UV technology startup, preferable in zero chemical residual, max UV disinfection is the major option for water treatment than chlorination. UV does not alter water chemistry and its constituent. Nowadays, more compact and efficient UV technologies design has been successfully created to be used in the residential area, which not only applicable in the commercial as stated previously. UV is a physical process harnessing the power and energy of photons to effect destruction of pathogenic microorganisms and break apart chemical bonds of pollutant. In order for UV to be effective, the photons both have to be absorbed by the target pathogen or chemical and pack enough energy to cause a lasting photochemical effect. The high energy associated with short wavelength UV energy, primarily at 254 nanometer, is absorbed by cellular RNA and DNA. This absorption of UV energy forms new bonds between adjacent nucleotides, creating double bonds or dimers. Dimerization of adjacent molecules, particularly thymine, is the most common photochemical damage. Formation of numerous thymine dimers in the DNA of bacteria and viruses prevent replication. In the treatment process, it can be shown that number of bacteria, viruses, molds, and diaspores cannot survive when exposed to the correct dose of UV light. Therefore, UV is considered as the best solution for water sterilization. And for the last literature review, the main UV sources for current ultraviolet disinfection system are low or medium pressure mercury lamp. These lamps are widely used in water treatment system. There are still many issues with them. The major concern is that the ultraviolet lamps are fragile and contain toxic mercury, which is hazardous to the environment and requires proper disposal. In the past few years, with the rapid development and improvement of the semiconductor industry, UV light-emitting diodes, known as UV LEDs, have emerged as a new source for UV radiation generation. An LED is a semiconductor device that utilizes semiconducting materials to create a p-n junction, which is hole and electron. The electron and hole recombine at the junction to emit radiation, and the wavelength of the radiation depends on the semiconducting materials. In addition to diversity in wavelength, UV LEDs possess several unique advantages such as environmental friendly, which is no mercury, compactness and robustness, faster startup time, potential less energy, and longer lifetime. The first two methodology for this project is the collimated beam apparatus setup and irradiation experiment. The experimental work we conducted using a bench scale, which is collimated beam apparatus that works in a batch flow to irradiate the samples in the study. This apparatus contains interchangeable low-pressure UV germicidal lamp emitting monochromatic UV radiation at 253 nanometer. The selected UV lamp is housed above a wooden box collimating tube 100 centimeters in length, with square opening and a manual shutter down the UV lamp. The distance between them is 10 centimeters and the shutter was used to allow and prevent light to the water. Sample water will be taken from the treatment plant DBKL Sentul located near UTMKL campus as a water source for the present study as a feedstock storage that located under the level of the UV reactor by a pump with 2 horsepower motor. It will separate wastewater samples by exposing to the light from the UV lamp with different doses by varying the length of the collimating tube and different exposure times. The fecal coliform, which is the microbe in the sample, was enumerated the exposure to UV lamp. A 100 milliliter wastewater sample was poured into a 7 meter diameter glass crystallization disc containing a magnetic stirrer under the collimated beam. Next, for physicochemical parameters, samples of influent water were analyzed for a variety of water quality characteristics. For example, BOD5 was determined following the method of 5 days biochemical oxygen demand. COD was also determined that colorimetrically using sulfuric acid and closed reflux method. Ultraviolet transmittance at 254 nanometer was measured using a UV visible spectrophotometer and a quartz cuvette with a 1 cm path length. TSS, TDS, and VSS were measured by filtration method. All analysis was performed according to the standard method. For microbiological parameters, samples of influent and effluent water were analyzed for fecal coliform and were enumerated after serial dilution in phosphate buffer and plating onto endo-agar at 45.5 degrees Celsius for 24 hours. Streptococci were enumerated after serial dilution in phosphate buffer and plating onto M. enterococcus agar at 44.5 for 48 hours. Vibrio cholerae was enumerated after serial dilution in phosphate buffer and plating onto TCBS agar at 37 degrees Celsius for 24 hours. Salmonella and Shigella also were enumerated after serial dilution in phosphate buffer and plating onto SS agar at 37 degrees Celsius for 24 hours. The last one is statistical analysis. Statistical software Minitab Release 15 was used to develop PCA, which is Principal Component Analysis, FA, which is Factor Analysis, and Analysis of Variance, ANOVA, of the experimental data. To get the expected result in this experiment, the efficiency of UV ultimately depends upon the number of bacteria associated with particles and the extent to which they are shielded. Because of the collimated beam test correct for the UV absorbance characteristic of the wastewater, the numbers may create adverse characteristic of the liquid itself, particularly low UV transmission. Potential clients for UV system may use the collimated beam curve for their particular wastewater to indicate its disinfectability. From the graph here, it will determine the fluence necessary to reach a target effluent count. Next, UV was able to remove 2 to 3 log removal of fecal coliform in aerated lagoon. Effluents at certain count. We'll see the difference in required doses for aerated lagoon. For the aerated lagoon effluent, it will interpreted by using statistical analysis principle, component analysis, and factor analysis. The main governing factors in this process, as deduced from the previous statistical analysis, were TSS, turbidity, UV dose, and COD for aerated lagoon effluent. That is all from me. Thank you for the kind attention.